Okay, so welcome everybody back to the last session of today. My name is Laura Hoffmeister. I'm part of the corporate R&D science engagement team at Bayer. It's a team that's working on overarching cross-divisional strategic R&D projects. I'm also part of the Bayer Bioethics office. So if you want to learn more about bioethics, stay tuned. There will be more information coming soon. So I think, we all probably are really excited about all this hot science that we learned about today. I think also quite overwhelming, really, really exciting. So there's, of course, great potential, possibilities of editing the code of life, even writing it. And these advancements in science, they bring us also continuously to those crossroads, where we ultimately, this leads us as a community, as a scientific community working in life sciences, but also as a society, come to very fundamental questions. Like, how do we want to live? And how do we want to shape life, actually? And so I think while most of us would probably claim we have a really good moral compass, ethics, here also the topic of today's talk, really brings additionally inter- and transdisciplinary discussion to the table, which is really important. And with this, I'm very honored to introduce to you our next speaker, Professor Anne Muigai from Kenya, from Nairobi, Professor of Genetics, Deputy Vice President, Vice Chancellor of the Academic Affairs Research at the National Defense University, Kenya. She holds a couple of additional roles that I would like to mention here. So she was a member of the World Health Organization Advisory Committee on Governance and Oversight of Human Genome Editing between 2019 and 2021. She is currently a member of the Kenyan Commission for University Education, the African Technology and Innovations Incubators Advisory Committee, and she has been previously a member of the board of the Kenyan Plant Health and Inspection Services, and on the Animal Genetic Resource Advisory Group for Africa. And I think another very interesting role to mention is that she's currently also the chair of the African Biogenome Project, which I think some of you might also have heard about. She holds a master in plant science and a PhD in population genetics and molecular biology from the Jomo Kenyatta University of Agriculture and Technology in Kenya. And I think it's also a great pleasure to say that she's also, since May this year, a member of the Bayer Bioethics Council, which is an advisory group that consults Bayer also on bioethical questions. And with this, welcome to the stage, Anne.
Thank you. So thank you very much, Laura, for that invitation, and thank you, Bayer Foundation, for the invitation to be here. I think it would have been nice to start off with bioethics, but I think it's even excellent to end with bioethics after listening to all the science talks. And bioethics is a consideration that I'm sure we all have to go through as we are continuing with our research. So in consideration of ethics itself comes from the Greek word ethos, and it's a branch of philosophy that is concerned with human conduct, and especially with the behavior of individuals within a society. When you go into societies, you do find that societies tend to be homogeneous because behaviors are either encouraged or discouraged, and you find that people sort of tend to behave in a certain way. And this looks at the rightness or the wrongness, if you can use that word, and the judgments that need to be made at every critical point on whether this is right or this is wrong. But when you think about evolutions of societies, I think we've gotten to a stage now where ethics is inculcated into our way of life. So you naturally find yourself conforming to ethical standards that have been set up within societies. But in research and in science, as we are continuously pushing the boundaries, it's important for us to continuously ask ourselves these questions on whether we are really doing what is right and what is wrong, without necessarily infringing on science, and making sure that the science is allowed to continue, but it's allowed to continue in a way that allows for the rights and the dignity of the persons to be considered. And very, very many times you do find that the word ethics and morality are used interchangeably. But they're basically the same, because morality also thinks about what is wrong, what is right, just actions, unjust actions. But when you think about morality, morality is more to do with individual personal decisions. And ethics considers the rightness or wrongness within a societal context. So I could give you an example, like in Africa, before colonizations, it was morally right to be polygamous. But after colonization and civilization and development, it kind of became frowned upon, and now, after many, many decades, polygamy is something that is morally wrong. So you can have presence which are continuously on the change, depending on the societal acceptance or rejection of a particular idea. And this is where research comes in, because you have to have a different hat that you wear that is not the same one that is you consider in the societies when you come to research. Because not only are you thinking about whether an act is right or wrong, you also have to think about that within the context of the entire human race. So protecting the individual, protecting the society, protecting the environment, ensuring that animal rights, human rights, and the people who are involved in these communities, questions are put into it. And when you have listened to all the researchers who are speaking today, you realize that we are such passionate people as scientists that you do need another eye to have a look at your study, to be able to ask you those difficult questions that you can very easily gloss over. So it's important for us to ensure that science is, and recruitment of people into projects is voluntary, that there's no monetary inducement or promise of cure that induces people to voluntarily join, because then they've not made an informed decision. They've made a desperate decision to save their lives or save their life of their loved ones. And of course, you want to ensure that the patients or the individuals who are recruited into studies are informed of good consequences, but also possible adverse consequences that might come about. And safety. I think you've heard the word efficacy of Cas9 throughout the day. And joined to that word is the word safety. How safe is this research that you want to push into a particular society? So here, we really, really have to be key that the research that is being undertaken is designed in such a way to mitigate against any unintended effects, because you can never guarantee that a research will be 100% safe. But you want to ensure that there is enough information that can allow you to give a good percentage of certainty that there will be very, very minimal harm that will be carried out in the individual. But also you have to protect against unintended positive effects, because sometimes you only consider unintended negative effects. Maybe it will result in a disease progression, but you can also have an unintended positive effect that if undue consideration is not taken into the project, you'll never be able to replicate because you didn't know how you got that result. And it's an unintended effect that could be positive, it could also be negative, but it's also very difficult to control. And therefore, it's good that these considerations are taken into to ensure that they're in tandem with what is happening in societies. So Laura mentioned that I was part of the committee at WHO, the advisory committee that came up with the governance structures with regard to human genome editing. And when I talk about ethical considerations surrounding the gene editing, I think it's a good point to start from the human point, because this is the one that has unprecedented ethical questions that comes about, much, much more complicated than what you would find in the plant world or in the animal world. So human genome editing focuses on two types, mainly somatic or germline. Somatic cell human genome editing deals with postnatal somatic cell genome editing. Those are done on individuals who have been born, obviously, and who are suffering from a condition. And we've seen several cases of those types of studies happening here. But in crafting the genome governance structures, it was important to consider the frontier of science, which is also dealing with prenatal somatic cell human genome editing. And these are edits that are done in utero before the embryo is born in order to take care of some of those deliberating inherited genetic diseases that you want to correct before the individual has been born. And then germline human genome editing is mainly on two fronts. You have the one for reproduction, and this is the one that deals with doing gene edits in germlines, in either embryos, in gonads, ovaries or sperm, and these are allowed to continue to birth. So these individuals are born, and the one for not for reproduction is the one that is done mainly for research in research institutions, where the embryos are not allowed to proceed to full growth, and their growth is terminated in a particular place. Now, it's human genome editing for germline cells is pretty complicated, especially because you're talking about edits that will form the future generations, because those individuals will be born, their genomics will be in the population, and they will pass this on to their offspring. So it's a permanent change, and the ethics around whether we want a human genome to be modified and to remain permanent is an ethical question that is out there for consideration. So we've heard about efficacy when we're talking about CRISPR-Cas9. And in ethical terms, what we're talking about is it safe enough? Is it safe enough for science to confirm that this particular technology can actually be used with minimum effects? So some of the changes that you want to mitigate against are incorrect on-target events. So you have point mutations or gene knockouts in areas that you didn't want to have. You can also have unwanted insertions or deletions. So if you have genes that are deleted, your chromosome ends up having genes that are missing, and there could be effects of these deletions on the population. There are also cases of chromosome damage or chromosome rearrangements. Natural phenomena like translocations, which happen naturally in cells, could be induced even by CRISPR-Cas9. But when they are done on cells that have never had this occurring naturally, then you could have changes which have not been anticipated, and this could have health implications on the particular individuals. Some of the health implications could be that the cells could grow in different ways. You could have tumors beginning to commence, and there are very many other effects, the most serious of which would be a lethal change. If you have knockouts of several genes on an important chromosome, then that particular individual could actually die. And this is why it's important to look at the efficacy of some of the gene editing tools, and CRISPR-Cas9 has some of the editing tools that were not very efficient as the science was beginning, but these efficacies are continuing to improve as we go along. So with postnatal somatic human genome editing, some of the risks that are associated with this is that some of the changes will be completely undetected. If the change has no effect on the particular individual, then it is okay if the change is undetected. But excuse me, if this change has an effect that is producing some adverse phenotype in the particular individual, then you want to know what event led to this. And the assumption that we have is that we have the technology to be able to go back to these areas to look for these mutations, deletions, translocations. And you wouldn't know what you need until you need it. And this is one of the risks that is associated with postnatal somatic human genome editing. And that's why the lab tests and the lab trials are really, really important before you get to the point where you're actually doing this in vivo. So you need to know what genes will be affected or what to look for. You also need to understand gene expression, because it's in the expression of the genes you will be able to understand exactly where changes have occurred. Now, if these changes are occurring in millions of cells simultaneously, the technology and the processes that are in existence to date, as far as I know, are not efficient enough to be able to detect all these changes occurring simultaneously. And this poses some of the ethical questions about the efficacy of postnatal somatic human genome editing, especially where these factors are not considered beforehand. Again, another question is this whole issue of defining what is safe. How safe is safe? What are your borderlines? Is 10% safe or is it 80% safe? Are you going to allow the technology to continue if the efficacy rates are 90 or 80 or 85? And that's degree of safeness is the one that requires a broader definition. Some of the risks also include genetic mosaicism, where you have cells that have genome changes occurring in different cells. And this occurs especially when the embryo replicates before the repair happens. So when this happens, then you cannot be able to prevent genetic mosaicism happening. And again, the downstream effects of this on the individual are yet to be determined. So it's a fairly common event, but it's completely unwanted. And some of the experiments that are currently going on is to how to ensure that replication doesn't happen before the gene edits happens in order to prevent cases of mosaicism. And then when we talk about human genome editing, especially for reproduction on germline cells, it's important for us to consider whether it's possible really to be able to get informed consent. Are you able to get informed consent from an embryo? And of course, the answer is no. Are you able to get consent on future generations, whether they will want to have humans who are edited? What will that individual say about their rights? And will there be, discrimination, racism against individuals who are individuals who have different edits? And do we have to reconsider their human rights, and whether people will decide that they're less or more human than others? So these are some of the ethical considerations when you consider germline genome editing. When you talk about somatic cell human genome editing, that particular gene edit will pass away with that individual when that person dies. But with the one for germline will remain for centuries. So can the parents make the decision? Is it the parents to make the decision, or is it the researcher? Who is the owner of the embryo? Who actually, and do we own embryos? So those are some of the questions we ask. Can you as a parent say that you own a child? You can't. But when you think about these sort of questions, individuals actually say that parents make these decisions for their embryos all the time. They say, I will terminate, I will not terminate, I will take my child for this surgical procedure, and that's a decision that a parent is making. But remember that this parent is making that decision. It's an informed decision because currently, using the research, surgical, medical knowledge that we already have defined probabilities of success or failure. So when a parent is making that decision, they already know what the chances of survival or benefit are on their particular child. But it's even more complicated now when you bring in the future generations whose future edit and the future potential of that gene edit on that particular future individual is taken into consideration. So it's a different question altogether. So it is because of this that WHO and many organizations around the world have said we don't have the ability at the moment to allow for human genome editing on germline cells until some of these difficult questions are considered. But basically, even more importantly, is the question of safety, definition of safety and reduction of these unwarranted and unwanted genetic changes on the particular individual. The other question that we have to consider is actually the cost. Not only the cost of the procedure, but the cost of the procedure on the public healthcare systems and on the particular country. You see, at the beginning, we had introductions about, food for all, the development of science and technology for societal progression. So when you talk about doing gene editing, we cannot do gene editing for people who can afford it, because right now, the cost of gene therapies are ranging between $325,000 and $2.1 million US. So when you're developing therapies for people who will be able to purchase it at these costs, then basically what you're saying is that you're developing a therapy for the people who can afford. And that is actually another way of limiting access. So when we talk about developing medical solutions, we have to think about access. We have to think about the ability of people from all over the world to be able to access this. And this is especially true for countries from lower and middle income countries, because when you talk about some of these diseases, the burden of these diseases is actually higher in Africa and Asia. When you think about beta thalassemia, sickle cell anemia, and yet these patients will not be able to afford it, and neither will their countries consider it a priority. They have bigger priorities of climate change, drought, provision of education, for them to spend large chunks of public resources on gene therapies. So there's a moral issue there. We have to think about it now from a moral aspect. How can we be able to increase access of gene therapies to patients who will not be able to access it, not only because of the cost, but because of the location of where these research studies are found? Again, when you think about where these gene therapies are happening, most of these gene therapies are happening in the developing world. And patients who require them from the lower and middle income countries will need to access this by traveling and being able to find places where they can get these therapies. So it's not only about the cost. So the particular cost might be $2.1 million US, but when you think about the support that this patient needs to travel, so they probably need an ambulance to take them to the airport. They will need the carrier to allow them to travel. They'll probably need admission en route to this particular place. They will need to stay in that country for 9 months, 1 year, while the gene therapies are being repeated, and these carry other costs. So the burden on the particular patient is very, very high. So we have to think about the duration of the treatment versus the quality of life when you think about questions of access to gene therapies for patients in these particular areas. So this is a map depicting the total number of medical travelers from the UK over the period 2000, and the implications of this, of patients traveling for Medicare, not gene therapy, just general Medicare, on the cost on NHS. Because what happens, at the end of the day, this patient will come back into the country and will use public-funded resources for management. And what was very, very interesting about this, while we from the developing world are looking to go to the UK for treatment, patients in the UK are actually looking to go elsewhere, Asia, Americas, and France. And so this is not a question of only patients from the developing world. Even we in this world here are looking to go to other places which are more superior. So already the access of this is a question for this, and this is a question of medical tourism. So how do we mitigate against this? We mitigate against this by researching or investing in research in situ. So having more research projects happening in these countries, especially in places where there's a higher disease burden, will prevent these patients from traveling out of those countries because they have access to research clinical trials. And then we need to have more investment in clinical trials in low and middle income countries, especially in countries where you have a higher disease burden. So it means stepping out of your comfort zone and setting up, replicate clinical trials in these countries in order to be able to help patients around these regions. But also, we have to ensure that there's capacity building, because if all the research and technology base is present in the developed world, then the patients in the developing world will also always have a deficiency. So developing in capacity building, building collaborations, investing in those regions will ensure that the knowledge exists there, and then the patients can actually remain there and be treated there. And then, of course, because of the IPs surrounding gene therapies and actually patenting of these and licensing of these drugs outside these low and middle income countries, we need to think about new models of how we can develop generics versus gene therapies so that these can actually be at an affordable rate in these particular countries. So when we're thinking about now setting up research hubs in other countries, we have to think about domestic rules for researchers. And when you think about some of the laws that are already in existence, you have to also consider that there may be an ethical issue that some scientists might try to go around. So if there's a moratorium on doing a certain type of research in your home country, will that researcher be able to move to another jurisdiction where there's no moratorium and do that research? Is that ethically proper? Because there must be a reason why there's a moratorium in their home country. And is that particular researcher taking advantage of the lack of laws in another region so that they can go and do this research there? So there's always the issue of, are we building capacity or are we taking advantage of the lack of laws in other jurisdictions? So would it be wrong to travel to another jurisdiction to do this? And is it wrong for that researcher to go and recruit patients in low and middle income countries to do research that would be prohibited back home? So those are some of the other ethical questions. And what is the implication for funding? Because right now in the US, for instance, you cannot get public funding for germline genome editing research, but private funding is still available. And how are these funds being able to move around? Because you can actually pass these funds through a circuit and still be able to use public funded monies for research that would generally not be allowed. So if this happens, is this a malpractice? Is it ethical? Is it not ethical? And how can we use international laws to limit movement of researchers from one jurisdiction to another jurisdiction? But then also locally, how do domestic laws apply? How would a nation in a country that doesn't have the particular law, how would they protect their patients from traveling to other jurisdictions, or protect their patients from being included in practice that is generally not accepted outside? So again, we need to think about international collaborations, bodies like the United Nations, UNESCO, WHO, developing governance frameworks that could actually be applied in different regions. And this is one of the things that we attempted to do. So what about that particular researcher who has moved to do research in another jurisdiction, and would the law reach them there? So if you're a European, you travel to another country to do work that is not allowed in your home country, would you be prosecuted? Would you be liable for law? And this is where, again, we need to have very clear framework on the international boundaries, and whether we can have a framework where you can actually punish a researcher who's gone out of their jurisdiction to do something that is not allowed for them back home. And then again, insurance. If somebody has traveled to a country to do a gene therapy in Spain, for instance, and it's a perfectly legal, perfectly ethical type of clinical trial being included on, when they come home, will they be shunned from the public funded insurance mechanisms that are there? And then this patient then would not have the full benefit of the treatment that they had invested in to go for such a long time outside. So again, I work in a military university. We don't do any enhancements. But again, this is a question that has come up so many times, especially in the WHO committee. Is it possible for us to use gene edits to create a superhuman? And this goes beyond the use of gene editing and science to create better life, to solve better problems. What we can say is that, the line between medical health and health for enhancement is very, very small. If you create gene therapies that are able to treat Parkinson's disease, you will automatically understand how the brain works, and it is possible for you to use that to your advantage to create a soldier who can be able to make decisions that are beyond the realm of humanity. So how will this be modified? How will this be regulated? And how will we even know? How do we know what the military is working on? Because it's highly restrictive and very, very highly secret, and in the knowledge base is very, very small. So this is something that is very, very real and is with us. But beyond that, I come from Kenya, and Kenya is known for running. Okay, I'm not one of those who run, but there must be a gene somewhere that confers some advantage to some of our runners because they come from a particular region. If this is understood, is it possible to use this and transform this to other people? And would these individuals be able to compete fairly with other individuals who are non-genetically modified? So these are questions that are coming up around the ethics of enhancement. And would this be superhumans as humans or superhumans as humans or human as human? I don't know. So there are ethical questions about that. So when do we cross a line? And how do we ensure that we have very, very strong law that will prohibit this line from being crossed into dangerous and very, very murky waters? And then, of course, when you're talking about enhancement, we're talking about designer babies. Because for as long as you're able to treat some of those genetic diseases, and the better we understand chromosomes, putting up chromosomes, parents will come knocking on your door. They want to have a child who has, potential to become, a soccer player or a tennis player or name it, or a money-making machine or, the next Michael Jackson, who knows. So again, we need very, very strong regulation. And remember that we as a society are the ones who will be able to delineate this and ensure that what is wrong now remains wrong even in the future, and that this doesn't change as the society moves along. Of course, one of the things that we observed in line with stem cell research is a lack of regulation led to the proliferation of rogue clinics all over the world, proclaiming to do things from cosmetic surgery all the way to even treatment of very, very serious cancers and leukemias and you name it. And this is something, again, we have to be aware of. For as long as you come up with a new technology, all of a sudden there are people who are ready to make money out of it. From the back of their garages with very small equipments, can inject products into patients, promising very, very high returns. So usually these are located in countries where there's a cap in law, and usually it's in lower middle income countries where people are very desperate to get jobs in labs without really realizing that these labs are actually doing things which are unethically considered in other jurisdictions. So when we come to gene therapies, even as much as we're publishing, as much as we're having conferences about our research, remember there are other people who are mining this information to go set up rogue clinics in different jurisdictions around the world. So the mitigation around this is regional laws, international regulations, and also treaties between jurisdiction for extradition of individuals who are doing rogue science, and also soft laws that can ensure that there's enforcement and discouragement. So once people are able to see that I'll be extradited back home and tried for pushing this type of therapies which are not allowed back home, it naturally acts as a deterrent. So there should also be pronouncements internationally in fora, even this, that right now we should not do human genome editing for future generations until we completely understand what the potential and the implications of this for the future generations are. So the other thing about governance and regulations is the importance that these laws and violations to the laws are detected. And they can only be detected if we have very, very robust whistleblowing techniques. There must have been people who were aware that He Jiankui was doing what he was doing. But there must have been scientists, collaborators, and there are publications that have provided evidence that it might have been the case that there were people who knew what he was doing, but for one reason or another, were unable to say what was happening. So how can we support very, very robust whistleblowing mechanisms? And how will we be able to ensure that the standards for the control of acceptable risk is acceptable in more than one jurisdiction? So will parents be able to make decisions on management of pregnancies that have gone wrong because they're carrying a gene-edited baby? And how will this neonate who is about to be born, if they're born prematurely, will the public systems in those particular countries, will the doctors be allowed or even accept to be able to manage these particular conditions? So whistleblowing is really, really important, and it's important that clear mechanisms are involved. Number one, to ensure that there's a way in which these mishaps and crimes are reported without unduly exposing the whistleblower, and also providing support for the particular whistleblower. That's when we will get to know about studies that are being done under the table. And then clear whistleblowing regulation also ensures that there's a very transparent mechanisms in which investigations will be done and the results published. Again, this will act as a deterrent for people who are doing things that are against the law. I've focused a lot about human genome editing on humans, but when you think about research in edits in plants and animals, the ethical concerns oscillate around the same thing. We talk about animal welfare, we talk about risks and safety of animals, we talked about acceptability of the gene-edited plants and animals to humans. Will they cause allergies? We also have to think about societal, religious boundaries when you talk about transgenic animals. We have to talk about ethical considerations, especially with regard to environment and release of these plants and animals into the environment. And the whole thing about transgenics. Right now, there is a very, very big move to reject anything that's transgenic, and the laws are prohibiting or discouraging movement of and development of the science in transgenics in a very, very big way. But we can think about transgenics across similar species, across tomato species, across cows, without saying that you're mixing genetics from different species completely together. And then also remember that the idea there is that all transgenics, plants or animals, will immediately have an implication on human health. So that we have to be able to speak very, very publicly that it's actually safer to eat something than to be injected with it. I always say this because with COVID we saw the acceptance of the public to being injected with the COVID-19 vaccines, but people are very, very reluctant to eat the GMO tomato because they think that has a higher risk. But I think it's the other way around. So there's also a very, very high risk if the experiment fails, and it's also very difficult to predict human reactions if novel traits are being produced. So in conclusion, it's very, very important to use science and development for the betterment of our society without restricting developments in science. And this can only be done if we have very strong, robust ethical laws and regulations in place. And we also need scientists to engage outside the world, to listen to the society and make sure that they're beginning to solve the problems that the society is having, and their views, the stakeholder engagement, their views are being incorporated into science. There is a large acceptance of somatic cell therapy around the world, and this should be encouraged to be developed and continue with higher safety mechanisms being put into place. But there is very, very little acceptance of heritable human genome editing, and this needs further research to answer questions raised on efficacy, safety, and mitigations of risks. And I thank you all for your attention, and I am ready for questions. Thank you.